Welcome to the channel again. Time for disclaimer. Let's start the cell series. Topic: extracellular matrix. Every cell lies on a cell bed made up of complex array of biomolecules that supports the cells. This becomes the major non-cellular component of connective tissues called extracellular matrix, also called ECM. ECM is present in most connective tissues including cell bed, bone, tendon, cartilage, eyeball, lungs, blood vessel wall, skin, and nearly everywhere. Main function of ECM is to provide support and strength to cells and also helps in their functions. ECM has three major components: structural proteins, functional adhesive glycoproteins, and proteoglycan. Let's start with structural proteins of ECM. Protein constituting 25% of the human body and most important structural protein of ECM is collagen. Collagen has a rod-like structure with 3000 angstrom long and 15 angstrom thick. It has unique secondary structure with 3 alpha chains, each in left-handed helical conformation. All 3 chains super twist around each other as triple helix with right-handed helical conformation. Left-handed coil super twisted into right-handed coils becomes coiled coil or super helix, leading to unique secondary structure collagen triple helix. Each chain has 1000 amino acid residues with tripeptide sequences that gets repeated itself. Glycine occupies every third position in the groove and the repeat sequence is glycine X, Y, N number of times. Proline occupies hundreds of X position, hydroxyproline occupies hundreds of Y position. Other significant contributors are alanine and hydroxylysine. In ECM, collagen triple helixes gets associated laterally with displacement of each helix by 1/4. This quarter displacement is called as quarter staggered arrangement of collagen, and it is responsible for cross striations and banded appearance of collagen in specimens. Collagen undergoes extensive post-translational modifications while being incorporated into ECM. First, proline residues undergoes hydroxylation by prolyl hydroxylase to form hydroxyproline. Hydroxylation requires ascorbic acid, vitamin C. Proline and hydroxyproline confers rigidity to collagen. Next, lysine and hydroxylysine residues undergoes oxidation by lysyl oxidase. Oxidation happens both between and within triple helix units, creating covalent cross links. This is copper dependent. These stable covalent cross links provide tensile strength to collagen. Finally, hydroxylysine undergoes glycosylation by addition of galactose or galactosyl glucose. Glycosylation site is unique to collagen and it aids its interaction with other components of ECM. Three major post-translational modifications are hydroxylation, oxidation, and glycosylation. Another important structural protein is elastin. In tissues that undergo repeated physical deformations, elasticity and recoiling is needed. The stretchiness and extensibility is provided by the structural protein called elastin. Tropoelastin monomer is the precursor form of elastin and there is only one genetic type of elastin. Amino acids that make up 75% of elastin are glycine, valine, alanine, and proline, the GWAP sequence. Formation of elastin involves self-assembly of 4 tropoelastin monomers through co-asservation. This creates tetrafunctional crosslink between 3 modified lysine residues and 1 unmodified lysine called the desmosin crosslinks. Crosslinks mediated through lysyl oxidase makes them extremely stable with half-life of 70 years. In extracellular matrix, elastin as a fiber contains mature elastin at its core. Elastin core is encased by insoluble microfibril that acts as a scaffold for elastin deposition. Glycoprotein that makes up structural component of microfibrils are fibrillin. Thus, the microfibril scaffold encasing elastin takes a beads on string appearance. Let's see next ECM component, functional adhesive glycoprotein, starting with fibronectin. Fibronectin has two identical subunits joined by two disulfide bridges. Fibronectin interacts with the cell through fibronectin receptor. The receptor is an integrin type transmembrane receptor with alpha and beta subunits. Further interaction with the cell extends indirectly to the actin microfilaments in cytosol. Attachment proteins interacting with the actin are alpha actinin and actin filament capping protein AFCP. The attachment protein from fibronectin receptor end is through talin and through vinculin. Fibronectin interaction with fibronectin receptor is through RGD sequence. RGD stands for arginine, glycine and aspartate sequence. These interactions allow cell interior to communicate with the cell exterior of ECM, and thus fibronectin affects cell behavior including cell adhesion, cell communication and cell migration. Let's see another functional adhesive glycoprotein, laminin, present in the basal lamina. Basal lamina of glomerular basement membrane GBM contains type 4 collagen. Type 4 collagen interacts with an adhesive glycoprotein with three distinct polypeptide chains, alpha chain, beta chain and gamma chains linked together called laminin. Laminin takes across an elongated cruciform shape. It has binding sites for integrin cell surface receptor and entactin. Entactin, also called nidogen, is a major cell attachment protein with RGD sequence. In basal lamina, these laminin proteins lines up together to appear as palisades. Binding proteins helps laminin to establish cell communications at ease. And so, laminin helps in cell adhesion, cell shape, cell migration, and cell differentiation. Let's see the last component of ECM, the gel that holds the ECM together, proteoglycans. Distributed between various ECM components are highly branched structures called the proteoglycans. Proteoglycans are proteins covalently linked with glycans, glycosaminoglycans. Glycans constitute 95% while proteins constitute only 5% by weight of proteoglycans. There are various types of proteoglycans classified depending upon the protein, glycan, their tissue distribution, and the function it serves. Examples are agrican, syndecan, perlecan, biglycan, versican, beta-glycan, decoran, serglycin, and fibromodulin. Let's see the basic structure of proteoglycan found in cartilage, agrican. Proteoglycans take the appearance of a bottle brush. It contains hyaluronic acid as the stem of the bottle brush to which are attached the link proteins. Hinged at the branches of the link proteins or the core proteins to which the other glycosaminoglycans gets attached. Glycosaminoglycans behaves as polyanions that tend to attract polycations like sodium and potassium. It results in osmotic movement of water into proteoglycans which contributes to ECM turgor. And that concludes extracellular matrix or ECM. Knowledge is bliss, let you be blissful all life. And signing off for now.